What if I told you that we could use bacteria to fight cancer? Yes, the same bacteria in your gut, E. coli, that sometimes cause you food poisoning, could also be used to do some pretty interesting things. Scientists are now hacking deep into this.
Let me explain how scientists have modified E. coli genetically to deliver cancer killing signals right inside the tumor. Could this be the future of cancer treatment?
This interesting fact was discovered in 1884 by a physician named William Coley. In 1884, the patient's luck seemed to go from bad to worse. This patient had a rapidly growing cancer in his neck and then come down with an unrelated bacterial skin infection.
But soon, something unexpected happened. As he recovered from the bacterial infection, the cancer also began to recede. Now, the real story begins. Dr. William Coley tracked down the patient 7 years later and he investigated the patient. He observed no signs of cancer. So, he assumed that the bacterial cells has stimulated the immune system to get off the cancer.
Over a century later, in 2019, synthetic biologists have found an even better way to use E. coli by modifying them genetically by manipulating their DNA to produce certain molecules. These molecules include chemokines that trigger an immune response inside the patient's body by attracting T cells and dendritic cells to the tumor site. So, why should we pay attention to this method instead following traditional methods like radiation and chemotherapy?
It's because this method can affect the entire body and disrupt healthy tissues as well while killing malignant cells in this process. But some bacteria like E. coli can selectively grow inside tumors where they can safely multiply hidden from immune cells.
So, how do you think E. coli identify their target tumor cells without infecting healthy body tissues? These bacteria have been adapted to identify certain changes such as low temperature, low pH, low oxygen levels, and overproduction of certain molecules. These changes help E. coli to identify tumors while avoiding healthy tissues.
Bacterial circuits like synchronized lysis circuits introduced into the bacterial genome to allow them to deliver medicine into the tumor cells. Avoid healthy tissues. They produce drugs only within a tumor cells while growing.
Then, a kill switch causes bacteria to burst when they reach a critical population threshold. This both releases the medicine and also decreases their bacterial population. However, a certain percentage of bacteria remain alive to replenish the colony. Eventually, their numbers grow larger and larger to trigger the kill switch again. And then the cycle continues.
Unlike many other therapies, bacteria don't detect specific type of cancer, but rather all solid tumors. Safe probiotic bacteria lie within our gut. Also, they could detect and prevent diseases even before they show symptoms.
To wrap up, hacking cancer with E. coli is still in the primitive early stages, but it is a very good prime example of how innovative microbial genetics can be. At the end, this isn't a science fiction. It is happening right now as we talk. By involving E. coli, we could create a smarter, safer ways of curing cancer. And could E. coli be that?